Hello, everyone, and welcome to today's webinar, Simplifying Pathogen Detection, Isothermal Amplification for Point-of-Care Applications. This webinar is part of the 15th annual event in the Clinical Diagnostics and Research Virtual Event Series brought to you by LabRoots. I am Israel Gonzalez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to www.thermofisher.com. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our chat feature during the presentation. You will also notice that the speaker has provided a survey located at the left of your screen. This survey will be available during the live session and the on-demand period. In addition to asking questions, we encourage you to participate in 3 poll questions at the beginning, middle, and end of the speaker presentation. Click on your answer when the poll questions are displayed. I would like to present our speaker for today now, Dr. Nina Pollak, Research Fellow, Center for Bioinnovation, University of the Sunshine Coast. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Dr. Pollak, you may now begin your presentation.
Welcome, everyone, and thank you for joining today's webinar, Simplifying Pathogen Detection, Isothermal Amplification for Decentralized Testing Applications. I'm Dr. Nina Pollak, and I'm excited to explore how isothermal amplification can revolutionize pathogen detection, particularly for decentralized testing. With advancements in this field, we are able to move beyond labs and bring diagnostics closer to point-of-care settings, which is critical in timely disease management. Now let's get started on how these innovations are simplifying pathogen detection and shaping the future of diagnostics. Before we dive in, here's a quick look at our agenda. We'll start by comparing polymerase chain reaction with isothermal amplification. Then we'll explore different isothermal techniques, including loop-mediated isothermal amplification, or LAMP, rolling circle amplification, or RCA, and we'll cover recombinase polymerase amplification, or RPA. After that, I will share some real-world point-of-care applications using isothermal methods. We will also discuss important considerations in designing diagnostic tests for point-of-care use. Then I will highlight how BioCipher, an Australian biotech company, is driving innovation in this space. And finally, we will look ahead to the future of isothermal amplification systems and their potential. Now let me introduce myself. I'm currently working at the University of the Sunshine Coast. I hold a PhD in Biochemistry and Molecular Biology, a diploma in business, and I'm also an Associate Fellow of the Higher Education Academy, which acknowledges my contributions and commitment to teaching and learning. My research is driven by one global central goal: to alleviate the disease burden in both humans and animals. I focus on advancing biotechnology and synthetic biology tools to develop rapid, accurate diagnostic solutions. This includes applying novel multiplex diagnostic technology, enhancing detection methods for arboviruses, mainly mosquito-borne viruses, in low-resource settings, and even engineering vaccines to protect endangered species, such as the iconic Australian koala. Throughout my career, I've been passionate about bridging scientific innovation with practical applications, and that is why I work at the interface of academia and industry. I'm excited to share how isothermal amplification fits into that vision today. Isothermal amplification is rapidly gaining attention in the point-of-care diagnostics space due to its simplicity and speed compared to traditional methods. There are several key technologies within this family. The two earliest are nucleic acid sequence-based amplification and strand displacement amplification, developed in 1991 and 1992. This was followed by rolling circle amplification and then loop-mediated isothermal amplification and later on recombinase polymerase amplification. But there are lots of others. While PCR has remained the gold standard for DNA amplification since its development in 1985, these isothermal techniques offer significant advantages for decentralized testing, particularly when rapid on-site results are needed. The figure here highlights the growing interest in isothermal methods, showing the number of scientific publications on LAMP, RPA, and RCA from 2003 to 2023. Let's take a closer look at how different amplification systems compare. PCR, the traditional gold standard, requires cycling through different temperatures, which extends the process to about 1.5 to 2 hours. It uses 2 primers and detects DNA through fluorescence, producing discrete amplicons, which means each amplified DNA sequence is separate and distinct from the others. In contrast, isothermal amplification methods such as LAMP, RCA, and RPA operate at a constant temperature, eliminating the need for thermal cycling. LAMP works at 65 degrees, RCA operates between 30 to 42 degrees, and RPA ranges from 37 to 42 degrees. RPA is the fastest, taking just 5 to 20 minutes. LAMP typically completes within 20 to 40 minutes, and RCA takes a bit longer, at around 1 to 1.5 hours. When it comes to primer design, RCA is the simplest, requiring just 1 to 2 primers. This is followed by RPA, which uses 2 primers and a probe, and then LAMP, which is the most complex. It typically needs 4 to 6 primers. For the detection, all 3 methods can use fluorescence or colorimetric detection, which involves a visible color change indicating a positive result. LAMP and RPA are also compatible with lateral flow assays, making them ideal for decentralized testing. Additionally, LAMP can be detected by turbidity measurements, so the cloudiness of the solution, and RCA through electrochemical methods. Lastly, the amplicons produced vary between these 3 techniques. RPA generates discrete amplicons similar to PCR, while LAMP and RCA produce concatenameric amplicons, which in general means the amplified sequences are linked together in long repeating chains. Isothermal amplification has proven incredibly versatile, making it a powerful tool for detecting a wide variety of pathogens across different sample types. In terms of pathogens, these techniques can be applied to a broad spectrum. For example, viruses like dengue, Zika, SARS coronavirus, where rapid detection is crucial for controlling outbreaks. We can detect bacteria, such as Mycoplasma pneumonia or cholera, where early identification can prevent severe health complications. We also can detect protists responsible for diseases like malaria or plant pathogens like late blight, which can have devastating impacts on both human health and agriculture. On the other hand, there are also fungi that we can detect, and that's from common infections like athlete's foot to more niche targets like the potato spindle turboviroid, the later affecting crops worldwide. As for the sample types, isothermal amplification can be used with a wide range of materials. Water samples, for example, both drinking water and wastewater. We can monitor contamination or disease spread. On the other hand, we have biological samples, including blood and urine and other bodily fluids, for diagnosing human and animal diseases. We can test food samples, like meat or fish, to ensure food safety, and even surface swabs from equipment or utensils, allowing for environmental monitoring in healthcare or food processing settings.
We would appreciate your participation by answering a few polling questions. To record your answers, click the option on the screen. Our first polling question is, which isothermal amplification techniques are you familiar with? LAMP, RPA, RCA, WGA, SDA, EXPAR, TMA, HDA, or other. We'll give you a few seconds to select your answers. Nina, back to you.
Next, let's focus on loop-mediated isothermal amplification, short LAMP. It's a technique that's been gaining popularity in point-of-care diagnostics due to its speed and simplicity. How does it work? LAMP relies on the BSD DNA polymerase, an enzyme with strand displacement activity. This enzyme initiates the synthesis of DNA, while two of the specifically and specially designed primers form loop structures. These loops make the amplification process more efficient, allowing for rapid cycling of reactions. Why is LAMP so effective? It's fast and efficient, often delivering results within 20 minutes. With multiple primers targeting the sequence, it offers high specificity, reducing the likelihood of false positives. And crucially, it doesn't require sophisticated equipment, making it ideal for decentralized, resource-limited settings. However, like any method, LAMP has its challenges. Reagent stability can be an issue, particularly in varying environmental conditions. The primer design is a bit more complex. LAMP usually uses 4 to 6 primers, and finding the right sequences can be a bit tricky. Finally, while LAMP is great for single targets, detecting multiple targets in the same reaction presents a challenge, limiting its versatility in some applications. Next up is rolling circle amplification, or RCA, a technique known for its ability to produce large quantities of DNA from minimal starting material. How does it work? RCA utilizes both DNA ligase and DNA polymerase with strand displacement activity. The process begins with a padlock probe that binds to the target sequence and forms a circular template visibly at the top of the figure. The DNA polymerase then extends a primer around this circular template, repeatedly synthesizing a long single-stranded DNA concatemer. What makes RCA special? High sensitivity. RCA is particularly effective for detecting low abundance targets and can be used in next-generation sequencing. Amplification of large quantities is another advantage. So RCA can produce substantial amounts of DNA from very small amounts of starting material, which is valuable in various applications. RCA does come with its challenges. The amplification speed is one of them. The process takes several hours, making it slower compared to some of the other isothermal techniques. It has quite high reaction system requirements because it's using DNA ligase and it needs a specific reaction environment. And so therefore it can make RCA more demanding and costly. Multiple target detection is a problem similar to LAMP. Detecting multiple targets in one reaction can be very complex and challenging with RCA. Finally, let's explore recombinase polymerase amplification, or RPA. How does RPA work? RPA combines several key components: the recombinase, single-strand binding proteins, and BSD DNA polymerase. The process begins when the recombinase here in purple, binds to primers and scans the DNA for homologous sequences, shown in the top of the figure. This forms a D-loop structure, which is stabilized by single-strand binding proteins, depicted here in cyan. Next, the DNA polymerase, in light green, extends the primer, displacing the strand and leading to amplification. What makes RPA noteworthy? Definitely its speed and efficiency. RPA can complete amplification in just 5 to 20 minutes at temperatures around 37 to 42 degrees, making it one of the fastest isothermal techniques. It's very portable. It's ideal for point-of-care testing, as it doesn't require sophisticated equipment or thermal cycling. Just a battery-operated heating block will do the job. The ease of use is another advantage. RPA is simple and doesn't require the high temperatures needed for thermal denaturation that other techniques use. And that simplifies the process and makes it also more suitable for field settings. RPA, like all the others, does have some challenges. Reagent stability is one of them. Maintaining the stability of the reagents can be an issue, particularly in less controlled environments. Primer design is also a tiny bit more tricky. Longer primers and probes can be restrictive when selecting targets, and designing these primers can be a bit more complex. Another challenge is non-specific amplification. There is a risk of amplifying non-target sequences, which can affect the specificity of the results. Imagine a world where point-of-care testing is not just a luxury, but a standard for effective disease management. This vision is driven by a set of principles known as AFHEA, a framework developed by the World Health Organization back in 2003 to guide the creation of ideal diagnostic tests. The criteria were updated in 2019 from assured to reassured. So let's explore these reassured criteria. R stands for real-time connectivity. Picture a test that seamlessly integrates technology. It connects with your mobile phones or readers to instantly relay data. This real-time connection means that healthcare providers have immediate access to crucial information, making timely decisions a reality. E stands for ease of specimen collection. Imagine a test designed for non-invasive specimen collection. No more complicated procedures. Just a simple, user-friendly method that makes testing easier for everyone involved. A stands for affordable. The ideal test is not just effective, but also affordable. It's designed to be accessible both to individuals and to the broader health system, ensuring that cost doesn't become a barrier to effective diagnosis and treatment. S for sensitive. Sensitivity is always key to avoid false negatives. The best tests catch every relevant signal, ensuring that no case is missed and that the results are as accurate as possible. S for specific. Specificity is equally crucial. A test needs to avoid false positives, giving clear and accurate results that guide correct treatment choices. U for user-friendly. The test should be as simple as possible. It should require minimal training and be straightforward to use, allowing anyone to perform the test efficiently. R for rapid and robust. We know speed matters. The test should deliver results quickly, ideally within 15 minutes, 2 hours the most, ensuring that treatment can begin without delay. Plus, it should be robust enough to withstand the supply chain, including storage conditions. E for equipment-free or environmentally friendly. The ideal test doesn't require sophisticated equipment. It should be operable with minimal devices, perhaps powered by simple solar or battery options. And when it's done, disposal should be easy, with recyclable materials that are gentle on the environment. And D, for deliverable to end users. Finally, the ideal test must be accessible to those who need it most. It should reach the people who are most in need, no matter where they are. The reassured criteria set the standard for creating a point-of-care diagnostic test that not only meet high technical requirements, but also address real-world needs. By adhering to these principles, we can develop diagnostic tools that truly transform healthcare delivery, particularly through decentralized testing in areas where they are most needed. Follow this 6-step guide for selecting and implementing diagnostic tests effectively. So first, we want to define the test's purpose. Clarify the test's intended use, whether for detection, monitoring, or screening. Then review the market. With over 40,000 diagnostic products available globally pre-COVID, it's essential to evaluate and compare their features. 3. A certain regulatory approval. Ensure the test has the necessary regulatory certifications. 4. Assess diagnostic accuracy in ideal conditions. Check how well the test performs in controlled environments, and only then move on. Step 5. Evaluate accuracy in clinical practice. Confirm that the test's performance is reliable in real-world settings. And 6. Monitor performance in routine use. Continuously track the test's effectiveness and address any issues during regular use. By following these steps, you can ensure the diagnostic test is effective and reliable. Now I will move on to some examples. The first example is a real-time fluorometric LAMP assay to detect Chlamydia psittaci, a study funded by AgriFutures Australia and supported by the University of the Sunshine Coast. Chlamydia psittaci is an abortigenic pathogen that poses significant challenges to the equine industry, particularly affecting thoroughbred stakeholders. It also represents a zoonotic risk as it can cause respiratory illness in humans. To evaluate the effectiveness of our diagnostic methods, we use the Genie system to test real-time LAMP for Chlamydia psittaci detection. On the top left of the slide, you can see the amplification curves with C. psittaci in red, orange, and yellow, and the negative control in green. This real-time fluorometer allows us to monitor the amplification in real time, so we can see how the curve appears. Additionally, LAMP assays can be run on these real-time fluorometers and have this special function of offering a high-resolution melt curve at the end of each amplification run. This provides an additional confirmation of positive results. An example of this is shown on the bottom left, where the high-resolution melt curves for C. psittaci are again highlighted in red, orange, and yellow, with the negative control in green. The detection limit of the C. psittaci real-time LAMP was found to be equivalent to 1 genome copy per microliter of template. In our study, a total of 88 tissue swabs from equine abortion cases were tested with real-time LAMP and compared to a reference assay, demonstrating a sensitivity of 100% and a specificity of 97.5%. Now, if a real-time fluorometer like the Genie isn't available, colorimetric and cybergreen LAMP methods offer effective, equipment-free alternatives using just a heating block. At the top of the slide, you can see an example of an endpoint colorimetric LAMP. Here, positive samples turn yellow, while negative samples are red, allowing for straightforward visual interpretation. Below, there's the endpoint cybergreen LAMP. In this method, positive samples appear yellow and negative samples turn brown, also enabling easy visual assessment. In our tests, colorimetric and cybergreen lamp showed 94% and 97% congruence with quantitative PCR, compared to the 100% congruence achieved with the real-time lamp assay that I just presented in the previous slide. We also assessed the stability of these assays at 4 degrees over a 6-week period, as decentralized testing environments may not always support -20-degree storage. All 3 methods, real-time, colorimetric, and cybergreen lamp, demonstrated consistent amplification performance. Both colorimetric and cybergreen lamp provide accessible, specialized equipment-free options for pathogen detection, making them a valuable tool for decentralized testing. Now I want to give you an RPA example with dengue virus. Dengue fever, a severe mosquito-borne disease, has surged dramatically over the past 2 decades. From 2000 to 2019, global cases increased tenfold, rising from 500,000 cases to 5.2 million cases. Brazil alone reported 5.5 million infections in the first half of 2024. In response to the substantial health challenge that dengue poses, we developed 4 RT-RPA LFD tests, each tailored to detect one of the 4 dengue virus serotypes. So there are 4 serotypes. These tests are integrated into a novel field-friendly procedure that processes mosquito samples in just 10 minutes. The process involves crushing the mosquito with a simple pestle, followed by a dilution step, making it possible to perform sample preparation without advanced equipment or specialized RNA extraction kits. This is followed by RT-RPA, and then lateral flow strip detection, where 2 lines indicate a positive test result and 1 line shows a negative result. The tests were validated using experimentally infected mosquitoes. The lateral flow strips for dengue virus 2 are shown here, demonstrating 100% sensitivity. Dengue virus 1 and 2 also achieved 100% sensitivity, while dengue virus 4 demonstrated 92% sensitivity, accurately detecting 11 out of 12 mosquitoes. These rapid dengue tests represent a significant advancement for low-resource settings, offering an effective tool for monitoring dengue virus in mosquito population. Let's continue discussing these low-resource dengue tests, but this time we are focusing on clinical samples like blood, serum, and plasma. Traditional methods, including sample extraction and PCR, take around 2.5 hours, whereas our rapid dengue test delivers results in just 35 minutes. The process is simple. It involves a 1-step sample extraction, followed by dilution, isothermal amplification using RT-RPA, and then a readout on lateral flow strips. For instance, the dengue virus serotype 2 test demonstrated 100% sensitivity and 100% specificity in a set of 60 serum samples, of which 16 were positive. To provide full context, not all tests achieved the same level of performance. This was partly due to the sample availability, with dengue virus serotype 2 actively circulating at the time of testing. In contrast, some of the other samples had been stored longer, which likely contributed to the lower sensitivity. We also evaluated the test specificity against other closely related or co-circulating viruses, such as chikungunya, Japanese encephalitis, West Nile, Murray Valley encephalitis, yellow fever, and Zika, as shown in the table. Crucially, each serotype test was highly specific, detecting only its corresponding serotype without cross-reactivity with the other 3 dengue serotypes. This high specificity, combined with the rapid turnaround time, makes this test a highly effective tool for low resource settings, supporting more timely and efficient disease management with clinical sample matrices. These studies were funded by the Bill Melinda Gates Foundation, BioCipher, and the DMTC Medical Countermeasures Program, and supported by the University of the Sunshine Coast.
Our next poll question is, do you use isothermal amplification techniques, or INATs, in your research? A, yes, I use INATs regularly. B, no, it is not suitable for my research needs. Or C, not yet, but would like to try. I'll give you a few moments to select your answer. Thank you. Nina, back to you.
Now that I've shared a few real-world examples of diagnostics well suited for decentralized testing, let's shift focus to the key considerations for designing a point-of-care test, incorporating the reassured criteria I introduced earlier in this webinar. There are three crucial steps in the point-of-care test process: sample extraction, isothermal amplification, and a portable device format. Simple sample extraction ideally involves a one-step process, or in other words, a single reagent, instead of multiple steps, what I would call a reagent system, or a complex column-based multi-step extraction. For isothermal amplification, I recommend methods like LAMP and AFHEA, as they offer speed along with high sensitivity and specificity, making them ideal for point-of-care settings. AFHEA requires a bit too much time, however, is ideal when your starting material is very low. Regarding the portable device format, there are several options to consider. Lateral flow is a very simple format, and thanks to the COVID pandemic, we are all very familiar with its result interpretation. Microfluidics involves controlling small amounts of fluid in channels on a microscale, allowing for precise manipulation of samples, and this is often used in lab-on-a-chip devices for diagnostics. It may be the perfect solution to combine extraction, amplification, and lateral flow to make it a truly portable, simple device format. Origami is another innovative approach that uses folded paper to create multi-step diagnostic platforms, combining simplicity and low cost while enabling complex sample processing and detection in portable format. Let's now explore some additional key factors for designing an effective point-of-care diagnostic test. Sample extraction needs to be not just rapid, but also safe and easy. Ideally, we want a process that's robust in various temperature conditions and consists of a single straightforward step. Importantly, this should also inactivate the pathogen, ensuring the sample is safe to handle. When it comes to the isothermal amplification, speed is key. It should also include a standard or internal control to ensure reliable results. Using temperature-stable reagents like lyophilized options can enhance durability, and from a cost-effective standpoint, multiplexing, so detecting multiple targets at once, adds a lot of value without increasing complexity. For the device format, portability is crucial. Think of equipment-free solutions like lateral flow tests. Even if we bring in automation, it doesn't have to mean complexity. Robotic approaches can still offer portable and efficient testing solutions. Finally, the future of smartphone-enabled detection is incredibly promising. As the cartoon shows, the goal is to integrate microfluidics with biosensors or optical sensing directly on smartphones, making decentralized testing even more accessible and user-friendly. Imagine running complex diagnostics right from your phone. I'd like to wrap up this section by focusing on something that often comes last in test design discussions, but it's actually the very first step in the process: sample preparation. It is easy to overlook, but without effective and safe sample preparation, even the most advanced diagnostic tests can't deliver accurate results. Let's talk about sample preparation, and specifically a low-resource solution that has real potential for point-of-care testing: DNA-cipher reagent E. This innovative reagent comes from an Australian biotech company, Biocipher, and for full transparency, I'm one of the co-inventors on the patent. What's exciting about this solution is how it simplifies the entire process. Instead of long, complex workflows, you get faster testing with sample processing completed in just 5 to 10 minutes at room temperature. It's straightforward, a 3-step process: add, wait, and test. It also offers increased throughput. Because it inactivates pathogens, it reduces the need for biohazard level 2 environments, which means fewer bottlenecks and less reliance on expensive infrastructure like biohazard hoods. This opens up testing to more labs without requiring extensive upgrades. Now let's talk about its accuracy and effectiveness. In trials, DNA-cipher reagent E showed over 97% accuracy when compared with commercial sample processing methods. It's incredibly versatile, handling a broad range of pathogens, including bacteria, parasites, and viruses. And one of the most critical points is safety. DNA-cipher reagent E doesn't just work fast. It also inactivates pathogens like SARS coronavirus 2, making it safer for lab personnel. Plus, it's compatible with emerging technologies like microfluidics, automation, and even bleach disinfection. Finally, I have to mention that this is Australian made. It's developed and manufactured here in Australia. This reagent has a long shelf life, remaining stable at the temperatures from -20 degrees to 37 degrees for up to 12 months. This truly makes DNA-cipher reagent E an excellent solution for low-resource settings and point-of-care testing. This one-step solution for sample preparation has been applied across various pathogens and sample types, making it a game changer for different testing needs. It's been successfully used for dengue virus detection in serum, plasma, and mosquitoes, malaria, in detail Plasmodium falciparum detection in mosquitoes and blood, Zika virus detection in urine, Nipah and Hendra viruses detection in viral transport medium, prawn hepatopancreatic parvovirus detection in prawn tissue, and even for identifying insecticide resistance in mosquitoes. This flexibility demonstrates how DNA-cipher reagent E can handle a wide range of sample types and pathogens, further supporting its potential for point-of-care testing in diverse settings. Let's talk about one of the most critical features of DNA-cipher reagent E, its ability to inactivate pathogens. This makes it safer to conduct testing outside of traditional laboratory settings. For example, with SARS coronavirus 2, mixing just 1 part DNA-cipher reagent E with 5 parts virus inactivates the pathogen in less than 30 seconds, which you can see in this figure. However, with the higher dilution of 1 part reagent to 9 parts virus, it remains ineffective. This demonstrates how important the right concentration is to ensure safety during testing. DNA-cipher reagent E also effectively inactivates dengue, Zika, Nipah, Hendra viruses, as well as bacteria like E. coli and Klebsiella. To sum it up, TNA cipher reagent E is a game-changing one-step solution for rapid, safe, and accurate sample preparation, effectively inactivating a broad range of pathogens, enabling faster testing, and simplifying point-of-care diagnostics without the need for specialized equipment. Lastly, let's discuss the future and promise of isothermal amplification systems. Earlier, I mentioned multiplexing, or multiple target detection. With recombinase polymerase amplification, we've shown at the University of the Sunshine Coast how an intuitive display is possible on lateral flow strips. So we've successfully coupled multiplex RPA with a compact and easy-to-use format. As you can see in the microfluidics illustration, RT-RPA was successfully integrated with lateral flow strips on a single microfluidic chip for COVID-19 detection. Another key requirement for isothermal amplification systems is long-term stability across a broad temperature range. Lyophilization or freeze-drying offers an ideal solution. Many isothermal amplification components are already available in this form, also by Thermo Fisher. For example, here you can see lyophilized lamp reaction in bead format for pathogen.
Our final polling question is, would you like to be contacted by our team to learn more about isothermal amplification product offering? Yes or no? Thank you so much for participating in these poll questions.
Let's summarize the key takeaways from today's webinar on isothermal amplification for decentralized testing applications to simplify pathogen detection. LAMP is a highly specific, sensitive, and fast method, making it ideal for various detection formats. With lyophilized reagents, it's easy to distribute as simple test kits, but multiplexing in a single reaction is a challenge. RCA requires minimal starting material and works well for low abundance targets and next generation sequencing applications. While it performs well at higher temperatures, its longer reaction times limits its use for rapid pathogen detection. RPA is fast, sensitive, and versatile in terms of detection formats. Unlike LAMP, multiplexing is achievable in one reaction. However, while lyophilized reagents are available, they come with some storage restrictions. In conclusion, isothermal amplification technologies offer promising solutions for point-of-care diagnostics, each with its unique advantages and challenges that must be tailored to the specific needs of the application. I hope this webinar has expanded your understanding of isothermal amplification technologies and helped you see why these techniques are increasingly gaining attention in the point-of-care diagnostics space. Thank you for your time and attention, and I'll hand this now back.
Hello everyone, I am Gintara and I am a product manager from Thermo Fisher Scientific. Today I am excited to introduce you to our range of products for isothermal amplification techniques. Let's start with the loop-mediated isothermal amplification, or LAMP. We offer the standalone Lyo-ready BST DNA polymerase, which ensures the ability to optimize your LAMP or RT-LAMP reaction. We also have the superscript 4 RT-LAMP master mix, providing a convenient, all-in-one LAMP solution. Moving on to recombinase polymerase amplification, or RPA, we offer standalone reagents and the Lyo-ready RPA kit, which provides all required components for fast, sensitive, and specific RPA or RT-RPA reactions. For rolling circle amplification, or RCA, we offer the wild-type Phi29 DNA polymerase and the engineered Equify 29 DNA polymerase. EquiPhi29 DNA polymerase is superior to wild-type Phi29 DNA polymerase in terms of thermostability, reaction speed, product yield, and amplification bias. We also have EquiPhi29 DNA polymerase available in a kit format for a complete RCA solution. Now let's look at some key features that differentiate these technologies. The first feature is reaction temperature. Our isothermal amplification products can operate effectively not only within their optimal temperature range, but also across a broad spectrum of 35 to 75 degrees Celsius. Another feature is tolerance to inhibitors. Here I want to highlight that isothermal amplification techniques generally have higher resistance to various inhibitors compared to PCR. Regarding sensitivity, isothermal amplification methods like LAMP or RPA are capable of amplifying low copies of pathogen RNA or DNA. For RPA, we demonstrated that it can detect the target with sensitivity down to 5 copies. The last feature is reaction time. LAMP and RPA are rapid methods ideal for pathogen detection where reaction speed is crucial. RCA is also suitable for target detection, but its longer amplification time is less appealing to diagnostic assay developers. Instead, RCA has gained popularity in applications such as DNA preamplification for NGS or in template preparation prior to in vitro transcription, where high product yield is important. If you're using standalone isothermal amplification reagents, we also offer additional components including superscript 4 reverse transcriptase, RNase out recombinant ribonuclease inhibitor, primers, DNTPs, and more. If you want to learn more about all the technologies and products we offer, please visit thermofisher.com/isothermal.
Thank you, Dr. Pollak, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsor, Thermo Fisher Scientific, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until November 13th, 2025. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye, everyone.